made it out of the body. Now, how do they move from one host to the next? This is what we refer to as the mode of transmission. Okay, and so there are three different modes of transmission that we're going to be looking at, so we'll go through each of those. The contact transmission is the first one there that we're going to talk about. And contact transmission, when you're talking about that would be, it falls into either direct contact, right? That's through touching somebody, kissing somebody type of thing. So skin to skin, membranes to membranes, whatever you want to call it, right? As far as depending upon what body part is actually making contact, okay? The other is through droplet. Droplet, in this case, would be any time we're looking at less than or about 3 feet or less. Okay, that's actually would be considered contact transmission. So in that case, here we're talking about when you're standing close to somebody, within that 3 feet, and you're having a conversation, and the moisture from the mouth or the spittle. If somebody coughs or sneezes, right, and the moisture that comes out at that point in time. So that all falls into contact transmission. And then the last would be the indirect contact. Indirect contact transmission is where those pathogens are being spread from host to host through fomites. Okay, and fomites are inanimate objects that multiple people are likely to come into contact with. Toilet seats, door handles, faucets, handles, right? Tabletops, cell phones, that and so forth. Okay, would be what we refer to as a fomite. Vehicle transmission would be our second form of a mode of transmission. And within vehicle transmission, again, just like within contact transmission, there are types. We have the airborne. And you say, well, didn't we just mention that with droplets? In a way, yes. However, when we designated it as airborne if they travel more than those 3 feet. Okay, at that point, we've moved away from that contact transmission into vehicle transmission. The air is actually, the droplets are remaining suspended and moving through a little bit further of a distance, right? This aerosols can occur from various activities, right? They can be through coughing and sneezing if that moisture then travels further with the help of a current of air. It can be through water fountains. It can be through misters, like at the grocery store, when they are, the thunderstorm sounds and they then, mist the vegetables. It can be through the water parks with pools, right? Or those water features where the water's coming up the ground and the kids like to play it because they're not actually in water, right? I always I found a picture that I finally found something that I liked. I love actually the video of these things, but toilets are nasty for airborne transmission. Okay, so this is just demonstrating when you flush the toilet, it actually is showing you the little particles of water, the little mist of water that's going to come up out of the toilet every time you're flushing, okay? And so that would also then fall under the airborne. Waterborne is where we're talking about the use of water. So again, in this case, your lakes, your streams, the natural sources of water, right? And oftentimes, this is going to be important the means of spread for those gastrointestinal diseases. These are oftentimes your fecal to oral infections, right, are going to be through this route. beautiful spring water when you're out hiking, right? Crystal clear looking spring water is not something you want to drink. Because just because it looks clean does not mean it is actually clean. That same water source is being used by the animals in the vicinity that use it as a drinking water source also. And unlike us, where we tend to defecate and void, you know, in other locations, not generally while we're actually drinking or consuming foods, animals don't have that same, you know, requirements, and so they will oftentimes defecate and void right nearby. And then you also have the whole, you know, rainwater with the runoff into that water supply source. Okay. Foodborne, of course, transmission would be the spread of pathogens in and on foods. Okay, and generally this is something that we can have control over by simply ensuring that, you know, the food is properly processed, cooked, refrigerated. Okay, when it is when it's inadequately processed, then that's where we run into our concerns and issues. You know, making sure that when preparing food, you are washing your hands in advance of doing so. The cutting boards or surfaces you're working on, right? You don't want to mix those surfaces. So if you just finished processing, cutting up the chicken meat, you don't want to use that same for anything else prior to disinfecting it, right? And cleaning it appropriately. As far as cooking meats and so forth, you want to make sure appropriate temperatures are being met for the proper you know, control of those microorganisms. And then refrigeration, making sure that you refrigerate your food, your whatever you're going to store, put it in the refrigerator right away. Okay, hot containers can go into the refrigerator, right? And in fact, you want them. You don't want to let them cool at room temperature before you put it in the fridge. That's never a good idea, because the entire time it's cooling down, it is now at an optimal temperature to grow. So microorganisms will grow. Yes.
Does it matter if it's covered or not?
No, it doesn't matter. Yeah.
Can you explain how cross-contamination works, like raw chicken being stored in the same place as other things in the fridge?
Well, well, first of all, in the fridge is not where the contamination is occurring. It's usually occurring in advance. Okay, and as long as the food is cooked properly, right? Once the food is cooked, then you're looking at any handling of it afterwards, or anything that's in the air that makes contact. And thus where I say that even though you cover it and leave it out to cool on the surface, it doesn't matter. The contamination has occurred prior to covering, right? But the cross-contamination that we're talking about is, you know, in the refrigerator. I don't know if that's necessarily an issue unless it's uncovered. When they talk about cross-contamination, they're really talking about, like I said before, when you're preparing the food in the same vicinity, one type of food versus the other. And the biggest problem you run into would be your uncooked foods, such as your salads. Nowadays, with as much education as they put out there to ensure that you cook your meats to certain temperatures, most people are pretty good about that. Where they have found it, which is why they show this here. Notice they show a salad bar, and all you can eat is salad bar, and that is because all the things at the salad bar are uncooked, right? So once it's on there, it's not going to be removed. Versus if you are, say, you end up preparing, say, like green beans, and then you turn around and cook them. When you're cooking them, you're going to be killing off. You see them? So that's when they're usually when they're talking about cross-contamination, that's what they're talking about in preparation of the foods. In a meat packing or processing facility, the cross-contamination of concern is when they're cutting up the meats, right? They have to be very careful with the intestines. And so cross-contamination occurs if they get any nicks or cuts in the intestines, where a lot of those bacteria are actually found, and then that gets transferred over to the meat. Okay, so I mean, it just depends upon where, you know, in the process you're talking about of concern. The problem with, because I run into this a lot. My mother-in-law is now living in the house with us too, and my husband, and both of them will not put stuff in the refrigerator. Like an hour later, I'm still seeing the food from dinner sitting on the counter. I'm like, freaking A, just put it in the fridge. Because the thing is that, yes, you've cooked, but after you've cooked, it's still exposed to the air for a period of time while you're eating your dinner, right? It's still, so anything in the air that's there can land. So then you cover it while it's still hot, right? It's slowly dropping down in temperature, and during that time, that those microorganisms, many of them are that they, you know, the ones that we're concerned about, pathogens we're concerned about, grow well at body temperature, right? And that body temperature range is going to be in that during that time frame that's sitting out cooling. So it's best to get it in the refrigerator so it cools as fast as possible in that environment versus a slow cooling process in the or on the tabletop surface. And I run into a lot of, well, then you get the condensation on the surface of the lid, right? So wet, you know, it's not an issue. So, but that does come into play, is getting it into the fridge as early, as quick as possible.
Even if it's covered?
Even if, that's what she asked, even if it's covered.
Would cross-contamination come into play also if you had like a piece of raw chicken, you cut it on a cutting board, and then you put it on there, and then you put like something else uncooked on there?
That's right.
Or if you touched an uncooked chicken and then touched something
That's what I
else in
mentioned
your
earlier,
hand,
is
like.
that if you have a cutting board and you process your meats, and then you take that cutting board and
Like
you
from
use
the raw
same
product
cut-
to the cooked product, yeah.
Right, and you use that same cutting board to use to process. And then the other is that, you know, some people, when you say you go out and grill, you take out your plate of meat outside, right? And then you put your stuff onto the grill, you start cooking it, and then, you know, I mean, I'm hoping not anybody here, but some people aren't necessarily thinking the whole process through, so they've got the plate right there, they brought the meat out on, cooked the meat, then they put the meat back onto that same plate, right? And so, because it's right there, I mean, that's an extra trip back and forth and in and out of the, you know, and so that's a problem, right? Because that could then, now you've just cooked, you know, potentially just killed off everything that was on the meat, and then now you're putting it back onto a plate that it could be contaminated. So, all right, and then lastly, for vehicle transmission would be bodily fluids. And when there's talk about bodily fluids in this case, it's bodily fluids that have already left the body, right, and are now in the environment, right? So vomit, or say an accident victim where there's now blood that is on and somebody's going through the process of cleanup. There's where they're talking about, you know, or defecation, that type of thing, where they're talking about it as a vehicle transmission. Okay. All right. And then the third of the different vehicle transmissions would be the vector transmission. And when we look at the vector transmission here, so something else is moving the pathogen from one place to another. And there are, these vectors tend to be, you know, some sort of insect that moves from one place to another. And so you have different types of vectors. They fall into two categories. You have your mechanical and you have your biological. And you say, well, the biological, of course, are the living ones. No, that doesn't have to do with that. The term is not to say it's living versus not living. The mechanical vector is one in which the vector is just transporting, much like a bee transports pollen from flower to flower, okay? The mechanical vector is then a vector that, an animal that lands on the food, such as a fly, it's most commonly brought up as an example for a mechanical vector, that lands on the food, right? But where do flies generally go? To meats, if you have an animal, a dog specifically, you know, to feces, right? So where was the animal, where was that fly before it landed on your food, right? Probably lays on feces somewhere, right? So, and like I said, it's like a bee with pollen. The bacteria are on the feet of the fly, and then they land those appendages. Then they land onto your food and transfer over those organisms then to your food. Okay, so it makes everyone really excited to go take a picnic, right? Just make sure you keep your food covered all the time, right, when you're not eating. That's the mechanical. So oftentimes, like I said, you'd think of flies for that. A biological vector would be a vector where the pathogen actually has part of its life cycle in the vector. And so these are oftentimes your biting arthropods. So fleas, mosquitoes, ticks, okay? Because they take a blood meal, they get the pathogen actually grows and has part of the life cycle within that animal, and then it transports it bites another, right, individual, and during that bite transfers. So one of the, there's quite a few of them that one could list off. The mosquito, Plasmodium is the species that is the cause of malaria, is one of those. Around here, of course, we hear about the West Nile virus big time during certain times of the year when the mosquitoes are high, right? Again, that's a virus specifically, but it falls with the mosquito. In the case of the mosquito, say with the Plasmodium species, the sexual stages of that protozoan parasite is within the mosquito. So the mosquito takes a blood meal off of an infected individual. It takes the pathogen into its gut where it grows, then it moves from the gut to the salivary glands, and then when it takes the blood meal from the next host, when you get a mosquito bite, part of that process is it regurgitates a little bit of its saliva into you, and with it the pathogen. Okay, and that's how it is transferred. The cause of Lyme disease, right, is one in which is caused by the transfer, or that mode of transfer is from one host to the next is through flea bite. The tick bites. I'm pointing to the tick, not the flea, but tick bites. Okay. All right. So let's see. Now we move, so that covers our 3 modes of transmission, each having several categories that fall underneath it. All right. Then we have, we're going to be talking a little bit about epidemiology. Now, epidemiology is the study of the frequency and distribution of disease. And anything, and actually includes any other health-related items that can come into play of concern. Right, and in different areas such as, not just with microbiology, but anatomy, physiology, immunology, medicine in general, psychology, sociology. These are all going to come into play with epidemiology. There's a lot of statistics involved with epidemiology. The biggest area that we are going to be really looking at would be the analytical epidemiology, which seeks to determine the probable cause, the mode of transmission, and the methods of prevention. Okay, and it's very, epidemiology is going to be very important and very useful when we're looking at situations where, say, Robert Koch's postulates isn't going to be as helpful. It is observations that are compiled and usually during an event, right? And then statistical analysis being performed in order to identify. They'll maybe throw in some changes that they make in order to quarantine, isolate, and then they'll gather data and evaluate how, the effect that had. In fact, we all went through this with COVID, right? During COVID, at the very beginning, right, we don't know what's causing this. They were gathering data, and then, 1 week, from week to week, it seemed like they changed their rules and what they were wanting you to do, right? And that is because they, using the information, they said, okay, this is where we think things are happening, how it's being transmitted. So let's put these steps into place. As they gathered more data over that next week or 2 weeks, they found, well, this is working, right? And so then things would shift as to how and what they wanted you to do. It's very much a retrospective process, okay? Not as helpful with the immediate so much as looking at what's happened and trying to identify what happened at that point in time, right? We can make changes that might influence what's happening now, but again, still very much retrospective. The individual who first really brought in that idea of epidemiology was Snow. John Snow mapped that cholera epidemic in London in the mid-1800s, which then really led to the identification that, hey, the water supply was the source of the cholera epidemic that was happening. And so then he helped put into place and helped move with that idea of isolating that community to prevent it from spreading, right? And then from there, of course, also quarantining and trying to make sure within that community we could get it under control. Now, in epidemiology, we look at things in regards to occurrences and distribution. So occurrence, there are two terms for occurrence would be the incidence and the prevalence, okay? And the prevalence is the number or proportion of individuals with a particular illness in a given population at any given point in time, all right? So when we're looking at prevalence, we're looking at total numbers from that population at that given time that have that particular disease state, okay? Versus incident is the number of, or proportion of new cases in a given population during a given period of time. So basically the same definition, except incidence looks for new versus prevalence looks for total. And you say, well, why would that make a difference? Well, new cases and where we're at with new cases gives us an idea as to what's happening now, right, and that transmission that's occurring, okay, versus total. Total looks at we have all of these people that are infected, but that doesn't mean that they just got it. That doesn't mean we have a current situation of transmission going on. It means it's in the population, right? So the two are looked at in order to keep an eye as to, what's in the population, yes, total, but also what's going on now as far as how is it spreading. And then, so with that occurrence, we kind of want to look at, they'll look at where in the US or in the world things are happening, right? During what time of the year. In fact, with COVID, that's how they knew it was a different virus, right? Because we knew the influenza virus its peak time is during a specific time of the year. This was at a different time. Okay, so we knew that there is a shift or a change in when this is happening during the course of that calendar year. It'll also look at population. Who are the most susceptible individuals, right? Are they, how is it demonstrating in that population? And then the distribution, when we look at those disease distributions, then we can categorize a particular disease that is happening into different categories for distribution. Sporadic is these random, small outbreaks that don't seem to be linked to anything, okay? And that happens, George Smith goes over to the Mediterranean, comes back home, and like, a week or two later is sick with malaria. It's not common for that area. It just happens here or there that you might have an event or an incident that has occurred, right? But it's not linked to anything specific. Obviously, in this case, just somebody traveling, coming back with a illness. It's not surprising, okay? An epidemic is an endemic. Endemic is where it's a chronic occurrence in a geographical area. So that's the levels for that disease that we expect to see for that particular area, right? And many diseases, we expect to see a certain number in that population throughout the year. So like the flu, okay? We expect in a population to have a certain number of incidents arise in that population over the course of the year. But when the number of new cases fall outside of the expected, that's where it throws it into an epidemic, a sudden acute disease outbreak. That doesn't mean that there weren't people sick with that particular disease state. It just means that now we've moved out of what we expected to see, and we now have this very high number that we're not usually seeing at this point in time. At that point, it is an epidemic. And then it moves to a pandemic when it starts crossing continents. Okay? So it becomes worldwide. I mean, pandemic just references a worldwide epidemic. All right. So here is a graph from your text, okay, of the HIV prevalence and incidence in the U.S. Okay, and so the incidence is the blue, new HIV infections, and the prevalence is the red. That's the total. And you can see that they don't always follow the same, they're not going to follow the same pattern, right? In this case, you'll notice that the total number of people in the U.S. that have HIV is continuing to rise. So it rose for a period of time, seemed to stable off based on this data, and then starts rising again. Why might our totals, first of all, why might they have started stabling off? I've seen graphs where the graph looks a little bit different, so you actually see a dip. Keeping in mind the years here, 1980 to 2010. In the so we first started seeing HIV cases in the early 1980s. Okay, it was all new, right? And we saw a pretty quick incline after that. As when we hit about the 1990s, we're seeing it stable off. Okay. Well,
You might be able to have, you have the indicator. In this particular, the numbers about the same. And then it's like more because they don't have enough information about the virus.
that would work for new cases, and you are correct. That explains what's going on here. Initially, we had a big increase in new cases, and then it started dropping off, and that was due to, and has dropped off and remained pretty constant. We haven't been able to prevent any new cases, but we haven't seen big numbers of new cases, and that is definitely due to education. Okay, and understanding how it was spread, how we can prevent spreading, and educating the public. Huh?
And the behaviors.
And behaviors of individuals, absolutely, changing of those behaviors. But for the total cases, okay, well, when somebody dies, they're not on this chart anymore. They get taken off the chart. Those numbers get taken off the chart. So total cases started leveling off more probably due to the fact that people died. So we have people are continuing to contract and give new cases so that increase, but also people are dying. That makes about sense. You know, we got about 10 years later from when we first started seeing it, where we get that, you know, the more people dying. But then, why then do we see an increase as we then proceed about the mid-1990s?
People started living longer.
And why would they do that?
Medication.
There we go. Yep, exactly. So medications, effective medications, then came into play, and so we now see total number of cases are increasing, continuing to increase, even though new cases are remaining relatively consistent. Total cases are increasing, and that is because it's no longer necessarily a life sentence. You're not going to necessarily die at this point with on medications, taking medications properly. You can live at just as long of a lifespan as somebody who does not have the virus.
It can be undetected.
It can be detected earlier. Mm-hmm.
No, it can be undetected. People that have been in medication for a while.
Oh, you're right. No, because it is because it's kept under control. Correct. The medication is keeping it under control, which is why circulating in their bloodstream it is no longer detected. Yes, I see what you're saying. Yes. Mm-hmm.
It does.
It doesn't change the total number of people who have it though, because they still have it.
It'll just lower the new ones.
It doesn't necessarily even lower the new ones because transmission is still possible.
It's just less likely if it's only
It's less likely to, yes. Yeah. But these are all the things that epidemiology are, what they're looking at. Okay, and what they're trying to determine then how they can, how it's transmitted, how they can prevent transmission, right? Now, this table also from your text is one in which we have a disease state. So in this case, they're talking about due to influenza and pneumonia. All right. Now, in blue is the expected. Okay. I'm sorry. In black is the expected. Sorry. In black is the expected. In blue is what they're saying. Anything above this is an epidemic. Okay, it's that threshold. So again, going back to what I mentioned before, is endemic, the fact that diseases are chronic in a population. Flu is and pneumonia present in the population, period. Okay, and so we have in our population during the course of the year, and so this is several years worth, and we expect it to cycle up and down during that year. We expect the highest in the winter months as we transition from one year to the next, right? That's what we expect. So anytime we go way above the expected, that is when we reach our epidemic. So the blue line is just indicating what they are referring to as the threshold for that epidemic, right? We expect some variance from our black line expected, but too much variance off is a problem, okay? So in this charting of these particular disease state, you can see we had an epidemic right here because it goes way outside of our expected range. We had another epidemic here, okay? And we really had another one here. So just because it exists in the population is not the issue. It's the outside of our normal expected. That's when it reaches that epidemic level. Okay? As long as it's following that black line, that's the endemic. The fact that we expect those levels present in the population, and that's where we're expecting it to be. It does not mean people aren't getting sick and dying from it. It's happening, but not at a level that's out of the ordinary of expected. Now, the epidemiology, or the epidemiologists, excuse me, pull together the information. They get information. Where do they get their statistics from? They get their statistics from hospitals, doctors' offices, nursing homes, right? And there's a list of diseases that they track. And any facility dealing with health, if they run across any of those on this list, they need to report it, right? They have to report it to the health agencies. Now, there's a federal health agency, right? And the one that deals with infectious diseases is CDC, Centers for Disease Control. And the Centers for Disease Control puts out the Morbidity and Mortality Weekly Report, the MMWR, a weekly report, and it reads much like the stocks and bonds, like getting the stocks and bonds trade sheet and going through that. It'll list the disease. This is like a condensed format of it. It'll list the disease. It'll list every single location in the U.S. It'll list the total for the U.S. as a whole. It'll list geographically for, say, northeast, southeast, mid, etc., right? And then it'll list every single state individually. And it'll provide you with where the current week is at, the numbers for that current week, the numbers for the median for the previous 52 weeks, the maximum for that previous 52 weeks. It'll tell you where we were at the year before. Also, you can do those comparisons as to where we're at now, as to where we have been. Get a retrospective analysis. So this is just demonstrating a few of those, and like I said, in a condensed form. I mean, this is a nice little stocks and trades type of format for these various diseases, and is compiled by the CDC and produced, and provided so that one could find out what's going on in any given area of population. And in order to make it accurate and reflective of what's currently going on, it is very critical that those doctors' offices, hospitals, all are putting that information or sending that information on. That's on the federal level, okay? On the state level, you also have a Department of Health. And then, of course, many of your counties and or cities will also have their own Department of Health. Okay, so you kind of work your way. You provide it to this, they provide it to the next, and reporting on up to the regulatory agency in order to get this information. Sometimes certain diseases, they actually require you to send in a specimen so that they can serotype that particular, and in order to identify, what grouping it fell into. Okay, what strain are we even talking about? And see if there's any links between different areas too of a particular strain. So the healthcare associated infections is another kind of dealing with epidemiology, another area of concern and an area that is highly reviewed and regulated, not regulated, I guess, but monitored is probably the best way to say that, right? The healthcare associated infections, these are watched by the epidemiologists because of the fact that these are facilities where we do, first of all, people are sick are at these facilities, and we see people who are sick are compromised, so there's a higher chance, because they're around others that are sick, oftentimes by other organisms, right? Transmission can be higher, so they want to be monitoring to ensure that transmission is not increasing. Because a healthcare associated infection is an infection where the patient or person, doesn't even have to be the patient, it can be the doctor, nurse, a visitor, okay, or the patient, go into, or the people who clean the facilities, right? Provide food for the facilities, anybody who walks into the hospital, and then acquires an illness from the hospital setting. That is then your healthcare associated infection. And they have a lot of aggressive control measures in place to try to prevent these type of infections from happening, right? Probably the most effective means of preventing healthcare associated infections is washing hands, okay? Regular washing of hands. Now, the types of healthcare associated infections can be exogenous. That means that the pathogen was acquired from that healthcare environment itself. So from the outside coming into whoever to that individual who got sick. It can be endogenous. So a pathogen that arises from normal microbiota within the patient. So the patient's already, so if the patient's in there, they're already their immune system's already stressed, right? And then they acquire an infection from their own normal microbiota. It can be iatrogenic, which is where the procedure itself, something about the procedure results in the infection, okay? They went in, had some, a surgery done, and everything wasn't properly removed, right? And now they're, they are now ill from, get an infection from that process. Super infections, okay, are where, again, we've talked about this one, where the antimicrobial that they've used to treat one infection now results in a different infection that has arisen. Big money is spent by the hospital on this right here. The Department of Health comes in and audits the facilities in order to try to target anything that could increase your healthcare associated infections, right? So they are, every hospital, every nursing home, these type of, even a prison setting where you have many people together, they are regulated. They have people who come, auditors that come in that are looking forward, making sure all the regulations are being followed to prevent these healthcare associated infections. So lastly, with epidemiology, the public health agencies that, one of their jobs, of course, is to limit disease transmission. As I mentioned, we have in the U.S., they are on the federal level, the state level, the county level, oftentimes the city level, okay? Worldwide, you guys know what the public health agency worldwide is?
WHO.
Who, right?
World Health Organization.
The World Health Organization, okay? These groups, all of them, their jobs, their reason for existence is to limit disease transmission, right? So their job responsibilities include that enforcing cleanliness of water and food supplies, working to reduce disease vectors and reservoirs, establishing and enforcing immunization schedules, locating and treating individuals exposed to, infectious material, and then, if necessary, establishing isolation and quarantining individuals. Okay. Also, another big part of theirs was public health education. Okay, that covers everything for me, I think, for this unit. Which leads me to our jeopardy. We'll go through these, don't worry about the timing. In lab, we are done with gram staining, right? Except for one person here. 2 people here, I'm sorry, 2 people here. Okay. Everyone else is done with gram staining, which means that when we go to the lab, it will just be, what we're gonna be doing for the lab portion is together, we're gonna be going through the test results. So I'll be talking about the results, you'll be looking at your results and recording them. Once I'm done, I'm gonna give you the list of new test tubes to inoculate. Once you inoculate them, you're out the door, okay? So it's not going to be for you, but everyone else, it should be fairly decent, timing wise. So I know we still have about 25 minutes for the class for us to do this, but don't feel stressed, because even if we run a little bit over, it's not gonna be a big deal. There'll be plenty enough time for the lab. Don't forget when you come up, to write down your name on the sheet so you get credit for participation. Today, I'm going to switch it up by starting in the back. In my back, 2 corners, either side.
What?
I'm starting in the back.
No!
Yes. And yes. I'm switching it up a little bit.
I got comfortable feet last.
I know, but see, I have enough of these that the front people end up doing a few extra, like 1 extra. So now you guys in the back are just getting opportunity. Just like last time, the first time we'll have you come up so I can sign, but after that you just stand up so I know where we're at.
Do you want to pick up?
Epidemiology 100.
Epidemiology 100. All right. A blank is an epidemic that occurs simultaneously on more than one continent.
B.
Yes, B. Hold on, give me a second.
I'm so sorry. My whole butt is.
You got this.
Oh god. I'm waiting for day pick. Yep.
Easy enough.
We're gonna have to come again up here, so. We'll just come up this side.
All right.
I'm so tired.
We have one of these for one of you. Will, yes.
I treated it.
Yeah,
okay.
No, bro.
Okay, which one? Treating disease for 100?
Treating disease for So which of the following is an example of a chemotherapeutic agent?
C.
C.
Look at you!
You're making me nervous.
What up, Tuesday when I work from home, I know he's 11
You can choose. I'll choose. Disinfectants. But you can choose
Okay, disinfectant.
What, hold on, guys, I can't hear. Disinfect for what?
200.
200.
Sorry.
Which of the following is one of the older and more common means of microbial control?
A.
A.
It's close.
No, we're supposed to have 3 now. Isn't that 300? Huh?
Oh, you're right, I'm sorry.
Yeah.
They did the 200 one, thank you. Yeah. I was thinking the 100. All right.
What do you want to know? Should you 100 or? You want to do that one? The relationships with 100?
100 relationships. A symbiotic relationship is in which one member benefits significantly affecting the other, it's not, without significantly affecting the other member. I can't even read it. C, yes.
I was.
I can't even read it out loud without messing that
Right.
That is the quiz question.
I gave it easy. That is? Oh.
Huh? No, I said it's as bad as the quiz. I didn't even read the quiz question right. In my mind, I was facing the question
We gotta do it. You can pick.
Which means it'll come up again. I hope it.
You're gonna
No, no.
I can do the disinfectant.
Do whatever you want.
The 300.
The 300 for, which one, the disinfecting?
Yeah.
You're putting.
An autoclave is able to achieve temperatures higher than that of boiling water.
B.
B is correct.
Whatever you feel comfortable in. Do the 100 then. Something easy.
Do you want to just do 100?
Yeah.
Okay, disinfectants and antiseptics 100.
See.
Antiseptics are disinfectant chemicals applied to Anis- D is correct. Antiseptics. Get us somewhere, please. Get us somewhere, please.
I'll let you
We're
pick.
counting on you.
Oh.
Our relationship with Michael is 200.
For how much? Good job. Microorganisms that are able to colonize the skin or mucosal surfaces for a limited period of time without causing an infection or disease state for the host.
D.
Neither.
Oh, it's transient, I'm sorry.
And B.
No, I messed it up because it did say for.
You can pick.
I can pick?
You can
Are you
pick.
sure?
Yeah, I don't want to make any more decisions today.
Just don't do 100.
I'm done making decisions.
Sure?
Yes.
I need that one. Hopefully.
I'm gonna do microbial infection, let's do 500.
I
told you. I told you.
A microorganism that has lost its ability to produce a functional adhesion factor is referred to as a
D.
D
Yes.
I was prepared to go to
I didn't want to choose.
All right. You're just standing up is fine. At this point, everyone's signed, right?
Yeah.
Okay, so I'll grab this. Don't wanna forget that.
Now the pen is full of microbes.
That's okay. It's a phone mic. What are we looking at?
Go ahead, whatever you want.
I got 100 here. What is the term used to describe characteristics of a disease that are felt by the, you said?
Damn.
Yeah, that's right. It sounds like a question though, like, All right. What do you want with? Daniel, go ahead. And I can't hear you. What? Okay. Treating disease 200. All right. The benefit of semi-synthetic microbial drugs may be blank than natural antibiotics. Not me. Not A. E. They are all correct.
And I ended
All
up
right.
doing 45%.
We're gonna take it home. Take it home.
And
Don't take it too far.
Do 500.
Do a 500.
Risk it all. Risk it.
All right, epidemiology,
Oh.
5.
whose responsibility is it to control disease transmission and spread?
B.
All of it.
That is correct. Disinfectants? Oxidizing agents are particularly effective against? B. B is correct. You close out the close out the disinfectants?
Sure.
It's
500. Let's go.
The current standard test in the United States for measuring efficiency of what?
D.
You said D, yes. But she did say first, right? I heard D first. I didn't even get to finish reading my question.
And you didn't give me my ring? How dare you? But what? No G?
All right. Treating for 300. Oxyzillinoids work to what? A is correct.
You got this. 500. Risk it all.
All right, let's do microbial infection 300.
300, right? Stage of infection recognized for mild signs and symptoms that precedes the more serious period of illness.
C.
C is correct.
Your turn, Leo. It's your turn, Leo.
Me? Oh, dang.
We started off so well.
Treating disease is 400.
Treating disease is 400. Which of the following is not an action that can lead to antimicrobial resistance? D is correct.
Is that right? I think you should read the answers.
I can't even finish reading the question. I think someone's had access to my computer or something.
Wait a minute.
You want to pick?
What do we got?
Epi for 4.
Happy for 4. Okay. The careful examination of data used to identify a cause or mode of transmission following an outbreak of disease is called what epidemiology?
C.
I heard B.
E.
You said E. You said,
C.
okay, well, you should have just said yes for my B, because I heard B.
I
But that
said
B
B.
was the correct answer. You said B?
No, I
No,
said
you said C. I thought she said
I thought
B,
she
and
said
she
B.
said
She
E.
said E.
I swore I heard B. All right, so we got, whoa, let me get back here. What do we got?
He's like,
What'd you say?
Treating the disease for 500. All right, that's what I heard. The antimicrobial polymyxin is primarily effective for treating superficial infections of
D.
what?
D.
Okay, not D.
Is it
She's like,
it's A.
F.
huh? Huh?
Wait, what time is it?
Wait, what?
Wait.
Polymyxin?
Hold on. Hold on. You said D.
Yeah,
Yeah.
I said D.
Yeah.
And I said D.
She did say D.
She did say
Okay. I'm sorry. D is correct.
Oh.
I'm sorry. I'm messing up my ears.
Out of your messenger.
I'm messing with you guys. I'm messing you guys up. No, D is correct.
Queen assistant.
Cut the camera.
Whatever.
Hazel.
Sure.
Huh?
Epidemiology for 300, he said.
A hospital patient acquires an infection during their stay. The lab determines the source of the pathogen was the patient's own normal microbiota. This type of an HAI is known as A. Endogenes.
Go ahead.
Jeez.
Listen, I was trying to read. Whatever you want. Please.
Let's go with microbial infection.
200, okay? Which of the following types of toxins are produced only by gram-negative bacteria?
A.
A is correct. All right.
Oh, whatever you want, girl. I don't.
Oh, micro-infected.
Last of the four.
Those are all.
When a housefly walks across feces containing salmonella and then walks across food, the fly is serving as a
B.
If it's not B, if it's not B, E.
E.
If it's not B, it has to be.
She was staring right at me, so I
Those are the only two that were there.
Which one? The 200? The last two?
It helped, right?
She was staring right at me, and I
For epidemiology, the incident is the number of blank in a given area during a given period of time. C. It was said by Okay, thank you. C is correct.
No? We up there, whatever.
Alright, which one? What is it? 300? Alright. Humans acquire normal microbiota in all of the following ways except.
In the womb, D.
That is correct.
Okay, ma'am. What?
There's not even enough.
Alright, so you're up, and you're, you guys.
Oh.
4 or 5?
4.
Okay. What can cause normal microbiota to become opportunistic pathogens?
All the above.
All of the above.
You know, we're doing better than that first time we did it.
Each time we get
A little bit smarter, you know?
Alright, ready? Which area of the body is not known to have normal microbiota? D is correct. Yeah. Page 2.